We're going to pick up where we left off. Hopefully you watched the podcast lecture on Tuesday. I want to pick up what we discussed on Tuesday, looking at protein function. Specifically, we're going to be talking about how enzymes work. How do enzymes catalyze key reactions in the cell, and so on. We're going to start, though, by talking about some measures of enzyme activity. And then we'll talk about regulation of enzymes, and then we'll get into the mechanisms, and then we'll close with mutagenesis and engineering. OK, so what we talked about on Tuesday is that proteins have a wide range of roles: structural, binding, and catalytic roles. In the example of structural, we saw, for example, collagen. And we saw how collagen gets organized into these complex assemblies that make it possible to have extremely strong bones and things like that. We also talked a little bit about titin, a muscle protein that makes muscles capable of being pulled and stretched without breaking, without snapping. We also talked about binding. The example we saw there was FKBP binding to FK506 and rapamycin. So, and then finally, we're now up to the part where we can start talking about catalysis. So protein function has at least three major roles inside the cell: structure, binding, and catalysis. And when we talked about the repeat proteins, for example, we saw a great example of binding. And I want to just very briefly, we'll take a look at that at a moment, but I just want to emphasize something based on some questions that I got during my office hours. We talked about how non-covalent receptor-ligand interactions can be described by dissociation constants and an on-rate and an off-rate. We're now at the point where we're going to start talking about this Michaelis-Menten constant, Km, which is an analog to Kd. And something that I touched on at the very end of the lecture is enzymes work by lowering the transition state enzyme of the reaction, energy of the reaction. Doing this is, in essence, how you catalyze a reaction. You're lowering the activation energy necessary for that reaction to take place. You're increasing the speed that the reaction takes place by doing this. And enzymes do this by binding to the transition state and stabilizing it. By having counterions that stabilize charge functionalities in the transition state, the enzyme can lower the transition state energy. And this is surprisingly effective. We're going to be seeing some examples of that today. So we're going to be talking today also about how this catalysis by enzymes is coupled to the motion by the enzyme. And I'll show you some examples of this. And what we're going to see is that enzymes force the substrate, the starting material, into conformations that favor formation of the product. And by doing that, that accelerates these reactions. OK, and very briefly, I just want to touch on one point about the repeat proteins. Something I didn't mention, but probably should have when I talked about the repeat proteins. This is an example of a repeat protein. This is an anchorin repeat. Notice that it has a series of helix, turn, helix, turn, helix, turn, helix, turn, helix, turn, helix, turn, helix. So those repeats, each one of these loops, and then helix, loop, and then helix. And notice that they're all stacked on top of each other. Each one of those is one example of a repeat. And this anchorin repeat has a whole bunch of these lined up. Okay, that's where it gets the name repeat protein. Make sense? And then similarly with the leucine-rich repeats, in this case, it's a helix, strand, loop, helix, strand, loop, helix, strand, loop, etc. Okay, but it's a series of a repeating motif, repeating structural motif. In fact, these are actually individual domains. They fold modestly well on their own, and they can be shuffled around to a limited extent. So, OK, any questions about this, the topic of protein structure? And for that matter, any questions about anything that was covered on Tuesday, in Tuesday's lecture? All right, well, I think in that case, then we're ready to move on. We're going to talk next about catalysis. Today's discussion is going to be pretty high level. I'm going to be telling you stuff that's actually not in the book. And in fact, actually, it's only found really in the frontiers of the literature in chemical biology. So don't hesitate to interrupt if at any point I start to lose you. Okay? It's better if you interrupt me early on than if I go further down the road and then you're totally lost. Okay, because the truth is, this lecture will be the only time that you're going to be able to find this material I'll be discussing, and I think it's very, very important. It's actually the very frontiers of chemical biology. Okay, so I want to talk to you today about catalysis. And last time I showed you that non-covalent binding consists of ligands hopping on to some binding site in a receptor. And we described this non-covalent interaction using an equilibrium constant, a special equilibrium constant called a dissociation constant that quantifies the ratio between unbound, up here, to bound receptor-ligand interactions. Okay, so this is pretty straightforward stuff. The only difference with catalysis is that we're going to have this similar receptor-ligand interaction, but upon binding to the ligand, the enzyme, in this case, instead of the receptor, and for that case, instead of the receptor, the ligand is going to be transformed, converted into some new product. So it's a similar sort of process. So if we understand non-covalent interactions, then we can also understand catalysis by enzymes. Okay, I'm skipping ahead. Skipping. We talked about this already. All right. I believe this is where we left off, right? Is this right? Okay. So where we left off, again, is this idea that receptor-ligand interactions are governed by some binding. In the case of enzymes, the substrate binds, and then some catalysis takes place to convert the substrate into a product. S stands for substrate, or starting material, and that gives us P, which is the product, and then the product has to dissociate. I think I've talked about this, right? This was covered. Right? I did cover this. Okay, good. Right. Okay, so I think I'm actually over here. Okay, good. So here's a typical reaction scheme I already talked about on the previous thing. So the formation of this enzyme substrate product, this Michaelis-Menten complex. This is sometimes called the Michaelis-Menten complex. This complex here of the activated enzyme bound to the substrate in a way to catalyze the reaction has an equilibrium constant. And in the same way that the Kd was proportional to the rate constants for the dissociation, the K off to K on, in the same way, the Michaelis-Menten equilibrium constant is equal to the sum of the off rate and the K cat. So that's like getting to the ES and either going backwards or forwards. That's the rates going in either direction for destruction of the complex versus forming the complex, meaning the rate constant for K on. Okay. This Km resembles the Kd for non-covalent binding interactions. And so it's useful for us. The strength of the Km tells us something about how avidly the enzyme is going to grab onto the substrate and how quickly it's going to form this ES complex. So this Km is actually a useful number. It tells us something about the conditions inside the cell, because the enzyme has to evolve up to a sufficient affinity for its substrate so it can grab onto the substrate inside the cell. If the affinity is too low, then the enzyme substrate complex will never form and the enzyme will never catalyze a reaction. On the other hand, if it evolves to the point where it's super duper high, maybe that's not so useful for the cell, because maybe the enzyme then will be a little too active. So these evolve up to the maximum ability that's necessary for the cell, that's required for the conditions found inside the cell. And oftentimes, for example, we can engineer enzymes to have very different Kms simply by tinkering with their active sites. And I'll talk about that more in a moment when we talk about protein engineering. Okay, so if we look at a dose-response diagram, this is similar to the dose-response diagrams that I showed on a previous slide on Tuesday. Instead, on the y-axis, we have our initial reaction velocity and the concentration of substrate. The point of inflection here is going to be roughly the Km, this equilibrium constant up here. For that matter, this point of inflection also tells us where 50% of the maximum rate of the enzyme is going to be. At the very highest concentrations of substrate, the enzyme is going to be running flat out. Okay? So that's like as fast as the enzyme can possibly go. It would be the equivalent of giving the sprinter maximum oxygen, maximum glucose, everything he or she needs to do to run as fast as possible. So that's up here under maximum velocity conditions. Notice that this asymptotically approaches this Vmax value way up here. Okay? And so anything where it's an excess concentration of substrate, that's called Vmax conditions. And typically, enzyme reactions that are run in the laboratory are run under those conditions. We always have an extreme excess of substrate, typically. OK, let's take a look at some KMs. They range widely. There's a wide range of possible KMs for enzymes. And here are some numbers over here. Now, like the KD, a lower Km value means tighter binding. Totally analogous to the dissociation constant. In fact, it has a very similar connotation. So what this tells us, for example, is that this enzyme here, cytochrome P450, binds benzopyrene with very high affinity. Okay, this should come as no surprise to us. Right? Benzopyrene is this big, flat hydrophobic molecule. And hydrophobic molecules in general aren't so soluble. So the cytochrome P450 in your liver is going to be grabbing on to the benzopyrenes that you inhaled on your way over here when you got behind that stupid shuttle bus that was, dinking along at too slow a speed. Right? So you get behind the exhaust pipe of that shuttle bus and you start inhaling unburnt benzopyrenes. Cytochrome P450 in your liver is right now, as we speaking, grabbing on with great affinity for these benzopyrenes. On the other hand, there are some enzymes that don't have to grab on all that well to their substrates, like a kinase. This is a key enzyme in the metabolism of glucose. And its substrate, citrate, is found at high enough concentrations that the enzyme doesn't really have to evolve to a very high affinity. So this gives us sort of a crude measurement of what the concentration of the substrate is in the cell. Right? So what we know is that there's probably not a lot of benzopyrene present, but there's probably tons of citrate present, hence the need for a lower affinity. Now, let's also take a look at some Kcats. So this is the rate constant for the decomposition of the Michaelis-Menten complex, the ES complex, that is now being broken down to form enzyme product. Okay, makes sense? Okay, so in this case, again, there's a wide range of Kcats. And this tells us something about how hard the reaction is to catalyze. Harder reactions in general have lower Kcats, but it can also tell us something about the evolution of the enzyme. Enzymes in general evolve up to the required function and really don't go past that. Okay, there's no evolutionary drive. There's no selection mechanism that drives the enzymes to be perfect unless they need to be perfect for some particularly crucial function for the cell. So here's one example of a really crucial function for the cell. The enzyme Catalase breaks down hydrogen peroxide into oxygen and water. This is a crucial reaction. Hydrogen peroxide creates a substantial burden on cells. This is a strong oxidant, and oxidants run around and wreak havoc on cellular machinery. And so for this reason, cells have evolved pretty sophisticated mechanisms to very quickly break down such oxidation products. And catalase has a Kcat of 100 million. So this is a really, really fast catalytic reaction that takes place. And then a slower reaction would be a protease. Proteases, of course, hydrolyze amide bonds. I believe we've seen these before. And their Kcats are much lower, likely because this reaction is a little more challenging and a little less favorable thermodynamically. And also, for that matter, it's not as critical, perhaps, for the cell. OK, questions so far? Good. Okay, so these are the numbers that are going to underlie our discussion as we start talking about the properties of enzymes. Okay, these are the same numbers that you learn about in like Bio 99 or 98, whatever biochemistry class you take here at UC Irvine or elsewhere. These numbers are kind of the vocabulary that my biochemistry friends talk about when they talk about enzymes. Okay, now the truth is, as a chemical biologist, I don't get too worked up about these numbers. I'm more interested in understanding the atoms and bonds basis for how the enzymes work. And so I guess the best place to start would be, let's start with the perfect enzyme. What would be the enzyme that really can crank, that could maximize its ability to turn over a reaction? And then we'll look at some specifics at the level of atoms and bonds. So the very perfect enzyme, you might imagine, every time it forms this Michaelis-Menten complex, the ES complex, then it goes immediately to Kcat. So it forms, and then boom, it's over to the Kcat. And it just immediately gets converts the substrate to the product. That happens instantly. On the other hand, the perfect enzyme is not going to have any off rate over here. This off rate represents lost opportunities. This is the the wasteland of coulda, shoulda, maybe should've. Okay, right? This is the chance that the enzyme missed. So instead of going to product, the enzyme goes backwards. And so this off rate over here is miserable and inefficient for an enzyme. So the perfect enzyme is not going to have an off rate. And so the perfect enzyme, you can basically imagine K off being 0. And if we have that, then we can imagine rearranging our Km equation, shown a couple of slides ago, such that K on equals the ratio of Kcat to Km. And again, notice that these little k's are indicating rate constant, and the big Km is for an equilibrium constant. So the very best enzyme will have an on rate that's diffusion controlled. In other words, it's limited by the amount of time that the substrate, Brownian motion style, eventually bounces its way to the active site. That should be the slowest step for an enzyme that's perfect. And we've talked about this before, but that rate of diffusion has a speed limit of 10 to the 9th per molar per second. Can't go any faster than that. That's a physical law. It's like the speed of light. You cannot exceed that, just because it takes a little while to bounce around through all that water and other stuff that's present in the cell. Okay, make sense? We'll take a look in a moment at an example of an enzyme that's far from perfect, and we'll start to understand what its sources of imperfection are. So before we do, let me just give you a little table that I really like that shows us and helps us organize enzymes. This shows us the rankings of enzymes in your proteome. Okay, so this is a listing from most common to least common. It's like a greatest hits of the 7 categories of enzymes that are found in the human proteome. The most common enzymes by far are the hydrolases. These are the enzymes that introduce water as a way of breaking a bond. And we're going to see a couple of examples of this. We'll see examples of glycosylases and proteases today. We've already seen examples of nucleases. That was stuff like RNase, right? Remember when we talked about RNase and it was inhibited by DIPSI? This is a similar sort of thing. Can someone help this guy out? Thank you. Jeez, please. Okay. Transferases, next most common, second position. These are examples of enzymes that transfer functionality from one spot to another. And we're going to look in detail at an example of a protein kinase today. And then later in the class, we'll look at a glycosyl transferase. Oxidoreductases, this is like the enzyme cytochrome P450 that takes the benzopyrenes introduces an epoxide and oxidizes the benzopyrenes. You all remember this, right? I showed you the benzopyrene a couple slides ago, but earlier in this quarter when I was talking about cigarette smoking, I showed you how the benzopyrene that looks like this rather innocuous flat structure gets converted into an epoxide and then slips into the pi stack of your DNA and alkylates the DNA. And so these are actually very common enzymes, these oxidoreductases, because they're important for removing toxins. Dehydrogenases are another one that's very common, and perhaps we'll get a chance to see this one today. And then finally we get down to the ligases. These are enzymes that spot weld together two functional groups, such as attaching ubiquitin or DNA to something. The isomerases are used to convert substrate into some related isomeric product. These are things like epimerases. The synthetases we've seen before. We talked about aminoacyl tRNA synthetase. This was that gargantuan complex that read out the anticodon and the various modifications of the tRNA to make sure that the correct amino acid was being attached to the tRNA during aminoacyl tRNA synthesis. Okay, so, and then the final one, the liases, are doing things like decarboxylation. These are actually breaking carbon-carbon bonds in dramatic fashion. So these are aldolases that are doing aldol reaction, et cetera. They're either breaking or making carbon-carbon bonds. So fairly, I feel like we've seen many examples of these different enzymes this quarter. So now I can go through and just talk about the ones that are really important that we haven't seen yet, okay, such as the kinases over here. And I believe that's where I'm going to start. Yes, in fact it is. So it turns out that kinases have a common fold that consists of a lower domain down here and then a larger lobe up here. The active site is indicated where these van der Waals spheres are. This is ATP. So kinases take ATP and transfer the gamma phosphate of ATP to some sort of hydroxide recipient. Okay, that's generically what they're doing. When we talk about the gamma phosphate, ATP, adenosine triphosphate, ATP has 3 phosphate groups called alpha, beta, and gamma. The third one in the row is called gamma. And so that's the phosphate group that the kinases are going to transfer. So again, notice that these have a conserved dual lobe structure, even though these have widely disparate activities. This is everything from a receptor tyrosine kinase over here to protein kinase over here. These do have very different activities. They phosphorylate different targets. And yet, on the other hand, they all evolved to have very similar structures. Now, this class of enzymes, like all enzymes, can be inhibited by substrates, pseudo-substrates, that mimic the real substrate. So here's the structure of ATP, but in place of one of the oxygens of ATP, highlighted in blue, we have a nitrogen. And this phosphoramide inhibits the kinase. Okay, so if you feed this phosphoramide to a kinase, to any of the kinases I showed on the previous slide, it's going to be game over for them. They're not going to be able to work because they're going to bind to this ATP analog. They're going to put it in a sloppy old bear hug, but this gamma phosphate is missing the oxygen. And missing the oxygen is the same as saying it's totally inert. And so this is going to basically be locked in the active site, in the brace of the active site, yet unable to transfer the phosphate group. And so the net effect is to shut down the enzyme. And this is a very effective way of killing enzymes. You basically know something about the mechanism, you make a tiny little modification of the substrate, and boom, it's game over for the enzyme. Okay, so you could do this also with sulfur or nitrogen. So I have shown you the nitrogen, but you could do that as well. And again, it sticks in the active site and inhibits the enzyme. This approach also works if you mimic the product. And a large number of enzyme inhibitors pursue one of those two approaches, either mimicking the substrate, as shown here, or mimicking the product. Either approach works great. So let's zoom in. I showed you the structure, the bilobe structure of kinase. Let's zoom in and take a look at its active site. In the active site, there are, here's the structure of ATP. There are a series of conserved magnesium ions, these balls over here, that are bound to the phosphate groups of the ATP. The numbers here indicate in angstrom the distances. Okay, and these numbers are pretty low, right? If you recall that a carbon-carbon bond is somewhere on the order of like 1.5 angstroms or so, these are pretty close in numbers, right? This magnesium is getting awfully close to this oxygen over here. These are cozy molecules and atoms. They like being this close. They like being this close because they have complementary charges, right? The magnesium has a +2 charge. The oxygens of these phosphate groups have negative charges. So they're attracted by salt bridges or Coulomb's interactions that we saw earlier in the quarter. So over here, there is the other substrate for this enzyme reaction that has a hydroxyl. I'm showing it to you with the hydroxyl deprotonated. And after phosphoryl transfer, this oxygen of the substrate has now picked up a phosphate group. And notice that the magnesiums here are helping to stabilize that phosphate group, right? That they're lowering the energy of binding by forming that same sort of Coulombic salt bridge that we saw earlier. Okay, now if we zoom in and take a look at the arrow pushing mechanism for this enzyme active site, what we find is that, and not depicted on this previous slide, somewhere out here, there's a carboxylate side chain from aspartic acid. The carboxylate of this aspartic acid deprotonates the proton of the hydroxy of a serine for the substrate, and that sets us up with an alkoxide. Alkoxide being a superb nucleophile, it's negatively charged, can attack the gamma phosphate of ATP, and again, the magnesiums get in on the action. They're over here participating fully and stabilizing this negative charge of the phosphate group. That's crucial, right? You can imagine this reaction not going in the absence of those magnesiums, right? Because one negative charge is not going to want to approach a negatively charged phosphate group, right? The negatively charged alkoxide over here is going to be stymied in its attack. It's going to get repelled by this phosphate group. So the magnesiums are shielding the phosphate group and protecting it and preventing it from getting looking like a negative charge. And so that tees up this reaction very neatly. And then finally, there's a collapse of this trigonal bipyramidal intermediate. And just very briefly, the structure around this phosphate looks like a trigonal bipyramid. Not so, anyway, that's interesting. And then there's collapse of this trigonal bipyramidal intermediate to give us our final product. Okay, so to summarize, the most important aspect of this is the notion that the magnesium ions are playing several roles to make this reaction possible. First, they're coordinating and stabilizing the transferred phosphate group as a Lewis acid. Okay, so that helps accelerate the reaction as a Lewis acid. Turns out that kinase activity in the cell is very tightly regulated. And the reasons for this are perhaps not clear if you don't know much about signal transduction, and I'm just going to very briefly cover it today, and then in a future lecture, we'll learn quite a bit more about it. In the cell, there's a series of pathways that transfer information, and these pathways are controlled by transfer of phosphate groups to key residues in proteins. So kinases play a really key role in kicking off various processes in the cell. So there's cascades of kinases where one kinase phosphorylates the next kinase, which phosphorylates the next kinase, and so on and so forth. Turns out that this process is very tightly regulated because you don't want your cells going wild. You don't want them to be doing uncontrolled cell division, for example. And so for this reason, the cell very tightly regulates kinase activity. And I want to show you a couple of vignettes about this tight regulation because it's crucial to our understanding of how kinases work. Okay, so here's one example. This is an example from the enzyme protein kinase A, a cyclic AMP-regulated kinase. And the way this works is there's actually a regulatory subunit shown here in blue that binds to the kinase and actually has an inhibitory loop that blocks access to the active site. Okay, so does everyone see how this dark blue thing is binding here, and then it has like this long finger that fits into the active site and blocks the kinase from binding to any substrates? This shuts down the kinase, and the ability to shut off the kinase is crucially important. Okay, if you don't have this, the kinase would be running around rampant, wreaking havoc, turning on stuff, shutting off stuff, causing death and destruction and general mayhem. And I do mean death and destruction. Okay, these kinases are that important. Now, when levels of a reporter molecule called cyclic AMP reach a certain concentration, this cyclic AMP binds to the regulatory subunit and causes the regulatory subunit to dissociate from the catalytic subunit of protein kinase A. So these two molecules get forced apart as the blue one flips into a new conformation. Upon binding to cyclic AMP, the thing changes its shape, and it no longer has affinity for protein kinase A. This is good news for protein kinase A. It frees it up to go off and do the mission that it's wanted to do for its entire life, which is to run around the cell and phosphorylate anything that moves. Nearly anything that moves. It actually has a little bit of specificity, but for the most part, protein kinase A likes to phosphorylate lots of different binding partners. Okay? This is a pretty promiscuous molecule. Now, here's the thing. Another way of regulating enzymes is to phosphorylate them. Okay, so this is one way where you have some regulatory protein that binds. A second way is to phosphorylate residues that are near the active site. Okay, so for example, this non-hydrolyzable analog of ATP, which has the nitrogen in place of the oxygen. This is the molecule I showed on a previous slide. This tells us where the active site is. But over here are two residues that can be phosphorylated to flip on this MAP kinase, this p38 gamma MAP kinase. And so one of these is a tyrosine, and the other one is a serine. And so this kinase waits around until it gets to the until it gets phosphorylated, and at that point it goes into gear. Okay, so this is like an on-off switch for the kinase. In the absence of this, the enzyme doesn't have the right conformation. It doesn't have the right conformation, it can't be a kinase. Okay, so the phosphorylation of the kinase puts it in gear, turns it on, and sets it going. Make sense? Any questions about what you've seen so far? Okay, that's the basics. I want to talk to you about the really neat stuff, the latest results in thinking about how kinases work and thinking about their motions. And again, this is kind of an abrupt departure from sort of standard material that's presented in biochemistry classes, and it really represents the frontier in chemical biology. Many of the next experiments I'll be talking about were done actually with Miriam. She's one of the leaders in this area. Okay, so the thing is, I want to talk to you about how enzymes work at a mechanistic level and how they actually work dynamically. How do they move when they do these reactions? So I should tell you that enzymes have great motions associated with their activities. This is unlike the case of conventional catalysis by organometallic complexes that you learned about back in Chem 51. Okay, or if you learned about in Chem 125. In those cases, the organometallic catalyst binds and perhaps it plays some Lewis acidic role, but it's certainly, we don't think about its motion. Okay, we don't think about it having some movement associated with it, some dynamics. Enzymes, it turns out, for the most part, almost all have very wild and very quick motions associated with them. And a frontier in chemical biology is to understand how those motions impact catalysis. How do those motions allow enzymes to be effective catalysts? And so it turns out that if you get a big, round bottom flask full of enzymes, you'll never be able to see those individual motions. And the reason is they tend to get blurred out. Okay, so if we look at a large number of molecules, we'll never see the individuals in motion because all of the enzymes in that flask are going to be running along at different speeds and everything gets blurred out. Okay, so instead, in order to see individual motions, we have to look at single molecules. And to understand this a little bit better, let me offer an analogy. Let's imagine that I convinced the Orange County Marathon folks to reroute the marathon. So instead of being on Sunday morning, instead I convinced them to run it here Thursday at 10:10 a.m. And I convinced them to start at that end of the classroom and send all the runners through the classroom from that door to this door. Okay, so now everyone's running through here, all 10,000 runners. You can imagine what you're going to see is just a blur of pumping arms and legs, right? Everyone's going to be trying to get in and out of this classroom as quickly as possible. It's going to be total pandemonium. That's the situation when we look at an ensemble of enzymes. It's total pandemonium. It's a blur of arms and legs. We don't see anything. Everything gets averaged out. And by see, I mean using tools like spectroscopy, using tools that you're familiar with from your other classes. So over the last 15 years, there's been a revolution in this area of chemical biology or biophysics where scientists have started to look at individual molecules in isolation from all of their other friends and neighbors. Okay, so now, instead of having the entire marathon coming through the classroom, let's imagine that I convince each runner to come running through one at a time. So they're going to start over there and then come running through here. What you will see, because each runner is isolated from all the other runners, is you'll be able to see their arms and legs moving, right? Because now there's no blurring out effect, right? And furthermore, if you look closely, you'll be able to see some runners moving faster than others. Maybe one runner has a different stride than her neighbor, right? Because she comes running through and I don't know, maybe she extends her leg a little longer than the runner behind her. Okay, but if we convince them to be isolated from each other, then we can really start to get information about how they move. And that's the situation we find ourselves in when we start looking at enzymes. And so this area of single molecules allows us to look at conformational steps, at intermediates, and the kinetics and dynamics that underlie enzyme function. And this, again, is a major frontier, and it's a really exciting area to be involved in research. Okay, so everyone with me so far? Everyone understands this idea of looking at single molecules, right? Okay, good. I want to talk to you next about how we're going to observe our single molecules. There are a number of different fluorescence techniques for looking at single molecules. Patch clamping, you may have heard of, is a 40-year-old proven technology that works really well for looking at individual receptors. That works fine. In the last 5 years or so, groups here at UC Irvine have been at the forefront for inventing a sort of a tiny little microphone that allows us to listen in to enzymes as they run. And it has some advantages over those other techniques. And so that's what I want to talk to you about today. Okay, so this is a collaboration between my laboratory and Phil Collins in the Department of Physics here at UC Irvine. And he's pioneered ways of building circuits that are based on carbon nanotubes. And this is an example of a carbon nanotube. This is basically a carbon graphite graphene layer that's folded up into a cylinder. It looks sort of like chicken wire. These wires, though, are amazingly conductive. Carbon nanotubes are a really remarkable material. They have remarkable mechanical properties. They have remarkable properties for conducting electricity and for conducting heat. In terms of conducting electricity, all of the electricity is going to be flowing through the outside of the wire, through these bonds out here. And the electricity is not flowing through the middle of the wire. So this is unlike, for example, the copper wires that are used in wiring the walls. Okay, wiring the electrical outlet over there on the walls. And this property makes the outside of the wire superbly sensitive to tiny little perturbations on its surface. And that's what we're going to do. So here's the way we do this. Students in the Collins laboratory and my laboratory start with silicon wafers that are about this big. We go to the engineering building across campus. The students put on bunny suits, and we build using photolithography circuits that look like this. There are these contact pads to which we attach wires. And then down here, you get down to these interdigitated electrodes that do not touch each other. So this is an open circuit. But somewhere out here, we sprinkle an iron molybdenum catalyst that catalyzes growth of one of these carbon nanotubes, a single-walled carbon nanotube, across the wires to complete the circuit. And I false colored it in red over here. OK, so now what we do next is we turn this wire into the world's tiniest microphone. We turn it into a device called a field effects transistor. It's not so important how that works. What matters is it's more or less the same as the microphone found in my cell phone. OK, same principle. And next, we're going to glue individual proteins directly onto the microphone and listen as they flap around. OK, so runners, if we had runners running through here, you'd expect to hear the pounding of their feet, right? And you'd expect to be able to interpret the noise of their feet to tell us something about their stride, whether or not they're accelerating, whether or not they're slowing down, et cetera, whether or not they have funny heel strike, et cetera, right? Make sense? So we're going to do the same thing, but with proteins. Now, I know that you're probably thinking, proteins don't have noise. I have proteins all over my body, and I'm not hearing anything right now. And the truth is, the noise is very, very tiny. It is so tiny that it's very hard to hear. But moving charges do make noise. And I'll give you one example of this. If you're at the beach with a bonfire down here at Little Corona Beach State Park, and you have this big bonfire going, you know how the wind kind of whips the flames, and the flames make this kind of neat whooshing noise? That sound of the flames moving is due to plasma in the flames. That's charged ions in the flames that are moving around. So charged functionalities make noise as they get pushed around. And in fact, actually, there's a speaker, a loudspeaker. These are really expensive stereo speakers, on the order of like $20,000 a pair. They better sound good at that price. But it's actually based upon having a plasma that's moved around by a little magnetic coil. OK, so you can actually hear charged ions moving around. And that's what we're going to do when we glue in the protein. So let me show you what it looks like when we have the protein glued in. This is the schematic diagram over here. We have the carbon nanotube. Here's the protein glued in. This protein is streptavidin, familiar, right, to everybody in this classroom. And we have streptavidin conjugated to a tiny little dot of gold, and that's shown here. OK, so the little dots here, that's the gold attached to streptavidin. And then the horizontal lines are the wires, the carbon nanotubes, and the vertical are the electrodes over here. And you can see we're getting one attachment. OK, so the breakthrough that Phil and I came up with our coworkers, our friends, the graduate students, was that we developed a way of attaching one and only one attachment each time to the carbon nanotube. This means then that we're isolating the enzymes away from all their buddies, which means then we can start looking at conformations and intermediates. OK, this is all a long introduction. We're going to get back to the kinases in a moment. Before I do, let me just set the stage. Here's the experiment again. We have the electrodes. We have the carbon nanotube. It turns out that, of course, you can't run your cell phone in water. So you can't run one of these tiny microphones in water. Okay, I think I did this experiment last week. I dropped my phone in a bucket of water. Actually, it's my cat's water bowl. And I pulled it out quickly enough, but it definitely did cause some damage. So electronics and water don't mix. I don't think it surprises anyone in the classroom. And so for what we have to do is but all biology, of course, takes place in water. So this creates a dichotomy. And to solve this, what we do is we cover up all the electronics with a layer of polymethyl methacrylate. This is shown here in gray. And then we blast a little tiny window using something called an electron beam that just opens up a little tiny region of the carbon nanotube. And that's where we're going to do the experiment. Now, all the images I've been showing you up till now are electron micrographs using electron microscopy. We're now going to get really small as we start imaging individual molecules of proteins. This is so small that you can't really see them very readily using electron microscopy, except if you use that trick that I showed on a previous slide where you coat things with gold. That was the streptavidin thing. So now to see these things, we now are going to have to use atomic force microscopy, where we're now getting down to really tiny resolutions of 1 nanometer or so. So here's what it looks like. This is one enzyme attached to the carbon nanotube. And this is an AFM image, atomic force microscopy image, just showing the windowed region, just showing the carbon nanotube that's exposed. This little blob right here is actually the enzyme attached. It has the right dimensions for that one enzyme. And now we turn on the microphone and it's lights, camera, action. Okay? At that point, then we're ready to listen in. Okay, one more image. This is before, and then that's after. And where it's circled, you can see very clearly the enzyme attached. Okay? Everyone still with me? Questions so far? All right. Yes?
What are the other?
Oh, thanks. These other blobs that are kind of large. Those are other little enzymes that we can't get rid of. It's actually enormously hard to do these sort of images. It just turns out the proteins are kind of sticky. There might even be some salt crystals somewhere out here. So there's always some garbagey stuff that we've been totally unable to get rid of, despite a lot of work. It took a lot of work to even get images that are this clear. So.
Could it be different when an enzyme get separate from others? The way they add reacts and the rate and stuff?
Okay, that's a good question. Okay, that's a really good question, actually. Thanks for not asking that. Thanks for not being on the study section that asked that. Okay, so the question is, would an enzyme in isolation from itself behave differently than an enzyme that's next to its neighbors? Right? In the same way that a crowded field of runners is going to run differently than if a solo runner. We don't know. I would like to think it's going to run the same. But it is a legitimate caveat, and I thank you for that. I will have to think about that some more. Thank you.
So why do we study that? Why we know in fact enzyme work in group instead of just.
Yeah, so enzymes frequently aren't really, so it's true, enzymes are in really crowded conditions inside the cell, but it's not like there's like 1,000 enzymes that are all doing the same thing crowded together. It's more like there's a couple enzymes that are kind of jammed in with hundreds of other molecules inside the cell. So they're not all doing the same thing. So we can recreate that sort of thing. We can recreate the crowded conditions inside the cell and compare. We haven't gotten to the experiment, but I'd love to do it. So, okay, let's get back to kinases. So again, this is protein kinase A. It still has the two lobes that I showed earlier. Here's the big lobe down here, and here's the smaller lobe up here. And somewhere close to this upper lobe, Miriam, together with Isa Moody, a former graduate student in the laboratory, engineered a single cysteine. The cysteine, of course, has a sulfur functionality, a thiol functionality, and that allows us to attach site-specifically the enzyme to the nanotube, to this microphone down here, through a pyrene. Okay, so pyrene is making yet another cameo in today's lecture. And as you might expect, the pyrene is going to pi-pi stack onto the carbon nanotube. Okay, because it's so hydrophobic, it's looking for a nanotube to stick onto. And it turns out the enzyme is very firmly held in place. This is like a very special kind of molecular glue that sticks these two molecules together. But notice that it's being held in a non-covalent interaction. And in practice, this thing is held in place for 10 or 12 hours. We don't see it coming off. It's really stuck in there very firmly. Okay, and again, here's another AFM image. And that little blob attached to the carbon nanotube is our enzyme. Okay? Yeah, question over here. Sergio.
How do we ensure that we have just one enzyme attached and not two?
Yeah, so we do AFM, this technique of atomic force microscopy, before we get started with the experiment, just to make sure that we have one attachment. And we use a special technique called in-liquid AFM. If we see two attached, we wash it away and then start over. If we see zero attached, we'll start over with attachment. Okay? Yeah, question over here?
So when you have one and you use AFM to ensure you have one.
Yeah.
Like, for example, a second one doesn't attach afterwards when you start this.
Okay, so afterwards, we don't add any more enzyme. So we have like purified buffer that has no enzyme around. So, and I know what you're thinking. You're wondering, well, what about this blob over here? What if it decides to get up and wander over here? It turns out these blobs are pretty firmly stuck down on the surface. And another thing is if another one attached, we'd hear that one running alongside it. Okay, right? In the same way that two runners would make different sound than one runner, right? You'd expect to hear a different rhythm. So we could detect that. We don't see it. Okay. Other questions? These are great questions, you guys. Yeah, over here.
Do you determine what enzyme you have based off of the AFM image?
We only add one enzyme, so we know that we have pKa around. Yeah, and then the AFM image confirms that we have one that's attached. Okay? All right, and question in the back, Anthony.
How do you actually measure the noise?
Okay, I'm getting to that. Okay, let me show you. Okay. Anthony is impatient. Okay, so you guys have exchanged songs by SoundCloud, right? So everyone, you raise your hand if you don't know what SoundCloud is. Okay, great. This is great. You guys are totally savvy. So when you do SoundCloud, you know how like there's always this little pulse that comes with it, and it tells you about the loudness of the thing. I'm going to be showing you images that are like that, data that's like that, where we're going to be watching noise. Okay, and that's shown here, where this is time on the x-axis, and on the y-axis, this is the current flowing through the nanotube. So that's going to be our noise. Okay, and enzyme by itself flutters around a little bit, but for the most part, it's totally quiet. Okay, so if you don't feed the runner, some oxygen or glucose, some gel, some bars or whatever, the runner doesn't start running. Okay, and enzymes are like that too. Okay, unless they get substrate, this enzyme happens to be totally quiet. Some enzymes we find kind of randomly flutter around. In our technique, we will not be able to pick that up. Okay, now when we add ATP, we see a new blip that appears. Do you see this lower blip over here? Each one of these corresponds to the ATP bound state. So we go from up here where the enzyme is open to down here where it's bound, and then back. And we can measure how long the enzyme spends in this bound state and derive a dissociation constant, a KD, for this enzyme, this pKa ATP interaction. When we do that, we find that that KD corresponds to what's measured in ensemble kinetics. That tells us that actually we're seeing something very similar to what's seen in the more crowded cases. Okay? Okay, next. We wash away the ATP and then add in a peptide that's a peptide substrate. This has the serine hydroxide that's going to be phosphorylated by the gamma phosphate of ATP. And again, we see some intermediate conformation as the enzyme goes from open to bound. Okay, and again, do you see how there's like more blips down here? The enzyme binds to its substrate, its peptide substrate, with greater affinity. It grabs on tighter. It has a lower KD. Okay, everyone still with me? Make sense? Okay, now check this out. This is the really cool one. This is now the enzyme plus ATP plus the peptide, and now we see 3 levels. Okay, so this is 1 second over here. This is now 0.2 of a second. I'm zooming in. And what we see is that these 3 levels correspond to ATP bound and then peptide bound, which then gives us a catalytically committed conformation. Okay, so when the enzyme starts working, it goes between open, intermediate, and catalysis. And I'm just going to call these 1, 2, 3. This is the waltz of pKa. Okay, so here's the enzyme waltzing, going along. 1, 2, 3, 1, 2, 3, 1, 2, 1, 2, 1, 2, 1, 2, 3, 1, 2, 1, 2, 1, 2, 1, 2, 3, 1, 2, stuck, 3. 1, 2, 3, 1, 2, 3, 1, 2, 3. So that's an enzyme in action. This is what the enzyme looks like as it goes about its business. Now, what's hugely inefficient is the K off. Do you remember earlier I told you the perfect enzyme should have 0 K off? In this case, we see the enzyme in real time, in action, being inefficient. Here it is being inefficient as it goes 1, 2, 1, 2, 1, 2. This is it with the K off. That catalytic inefficiency is what dooms the enzyme and makes it waste opportunities. The enzyme is trying to make up its mind. Go to product, back to substrate, product, substrate, product, substrate. And that lack of decision is what makes this enzyme inefficient as a catalyst. There are other kinases, such as kinases, involved in metabolism that are far, far more efficient than this enzyme over here. If you're looking for something, could I ask you just to wait until after the class is over? Oh, no, you're here for the class. You're just kind of late. OK, no problem. Welcome. All right, so anyway, this is the enzyme in action. And what this shows us is that the enzyme fluctuates its speed enormously. OK, and this is kind of mind-boggling. And I'm just going to tell it to you. It turns out that the enzyme's speed fluctuates from second to second by a factor of 100. Okay, so this is like going out to the 73 out here, the freeway out here, and then pulling up alongside a Honda Civic. And then suddenly the Honda Civic goes from 55 miles an hour to 5,500 miles an hour, and then back down to 50 miles an hour, all in the course of a second. So these enzymes are wildly changing their speeds. They're changing speeds much faster than any runner. They're changing speeds up to speeds that are wildly, that are almost inconceivable to us humans. And really, that's the stuff of life. It's essential that this enzyme is able to alter its speed in order for it to be regulated. You remember earlier, I talked about the regulation of protein kinase A. That regulation is going to control its speed, and in doing so, that turns the enzyme from being a wildly efficient catalyst to being a catalyst that's not even worthwhile, that doesn't operate on a time scale that's useful for the cell. And this is really the essence of how catalytic biology takes place. Okay, any questions about this? Yeah.
Do you see any pattern along that, the on-off, or it's just random process?
Okay, this is a brilliant question. We spent a lot of time looking for patterns in our data and looking for correlation between one step or another. And in short, it is pretty random. There's a small amount of a memory effect in the sense that the enzyme, if it hits hits this intermediate state, it's more likely to go down to 3 than it is to go back to 1. Okay, and so that's actually a thermodynamic effect, that the enzyme has evolved to do K-cat in preference to K-off. All right, let's talk about let's talk about a different enzyme. The second enzyme I want to talk to you about today is Lysozyme. This is an example of a hydrolase. And remember, I'm moving down, or actually I'm still at the top up here in our most common enzymes. This enzyme was discovered about 100 plus years ago, and it's been intensively studied. This is the X-ray crystal structure of lysozyme. And in fact, it was the very first enzyme X-ray crystal structure ever solved was this enzyme. The active site up here has an 8 angstrom hinge motion. And so this enzyme has kind of a Pac-Man-like motion as it hydrolyzes the glycosidic bonds of the polysaccharides found on the cell surface of bacteria cells. Okay, so here's a bacteria cell wall. And the enzyme is going to cut apart the glycosidic bonds between each one of these glycan moieties found on the cell wall. Okay, so it's going to be chopping apart the polysaccharide. In doing this, this will basically burst apart the cell. In the absence of this, the enzyme is basically going to be chewing apart the bacteria, and this has the effect of killing the bacteria, right? You're breaking their cell walls, they explode, et cetera. This enzyme is found in high concentration in chicken eggs. That's the hen egg whites over here. And it's present to prevent colonization by bacteria. Okay, and you might recall avidin was isolated also from chicken egg whites. So biochemists have been studying what makes eggs so special as sterile vessels for a very long time. Okay, let's take a quick look at the arrow pushing mechanism for how this enzyme operates. In this mechanism, the enzyme goes through a covalent intermediate. Let's start over here. So I'm zooming in now to the polysaccharide region of the cell wall of the bacteria. Enzyme is going to cleave this bond that's indicated with an arrow. And this is an example of a hydrolase, meaning it's going to introduce water across this bond. So the first thing that the enzyme does is torque this N-acetyl muramic acid moiety. Okay, so it's going to torque this carbohydrate from being a nice chair conformation to being a boat conformation. This is a crucial aspect for what makes enzymes such effective catalysts. This enzyme is going to be catalyzing a reaction 1,000 times more efficiently than if the reaction just had to happen by itself. I actually think it's like 100,000 times more efficiently. In order to do this, the enzyme is going to be physically bending the substrate. And by physically bending the substrate, this helps to accelerate the reaction. So here it is, pushed up into this boat conformation. Notice that the boat conformation neatly sets up an SN2 attack by this carboxylate of aspartic acid to attack backside displacement style this glycoside. Okay, this is crucial. So the glycoside gets protonated by one glutamic acid, and then over here an aspartic carboxylate from a nearby aspartic acid then attacks, doing a backside SN2 displacement. And this protonated glycosidic bond then is a very effective leaving group, because the second arrow, highlighted in red over here, kicks electrons to a positively charged oxygen, which is all too eager to accept those electrons. Okay, so this gives us a covalent intermediate. This is another common way that enzymes accelerate reactions. In this case, we're seeing it form a covalent intermediate, and then this covalent intermediate gets hydrolyzed. So half of the polysaccharide floats away, the next one comes, water comes in, gets deprotonated by the glutamic acid, the glutamate up here, and then this hydrolyzes the ester bond between this saccharide, this polysaccharide, and the aspartic acid. Okay, so some notable features here. I'm showing you an example of acid-base catalysis. The enzyme is simultaneously acting as both an acid over here and as a base over here. In fact, it's even wilder than that. Check this out. It's the same functionality, this glutamic acid, that acts as both the acid and the base. And to me, that's just an elegant simplicity that makes enzymes so beguiling. Right? If we were in the chemical laboratory trying to make molecules, using glassware and such, we'd either dump in a bunch of acid or dump in a bunch of base, but you wouldn't add simultaneously both acid and base because they would neutralize each other. And enzymes have evolved to be able to simultaneously catalyze things using both acid and base catalysis. Furthermore, this enzyme has evolved to form a covalent intermediate. Sometimes this is referred to as a ping pong mechanism that eventually gives us back the hydrolyzed glycosidic bond. Okay, really, really beautiful. This is what you learn in biochemistry classes. And the problem with it is that it neglects enzyme dynamics, which are really critical. As the enzyme moves, it can then help to torque this conformation of the ring into this boat conformation. In the absence of this movement, it doesn't make sense really why it is that the enzyme is actually going to be torquing this substrate, right? Because the substrate binds in the chair conformation up here. Why should it get pushed into this other conformation unless the enzyme is doing the pushing? And that, in fact, is what we see. Okay, so same idea. We're going to attach an enzyme to the carbon nanotube and then listen in as the enzyme works. When we do that, here's a paper from the lab from about a year ago. What we see is that, again, the enzyme by itself is relatively quiet. And then when we add the substrate, the polysaccharide, the peptidoglycan that I showed on an earlier slide, the cross-linked net, there's an immediate jump upward, and then there's all this noise in here. This is the enzyme chewing on its substrate, and we get to listen in. It's just like SoundCloud, basically. Okay. Some controls. I haven't been showing you controls, but these are everything in biology. This is substrate by itself in red, overlaid. And then this is enzyme by itself. Again, it's relatively flat in purple. And then when we have enzyme plus substrate, we see this motion where what we're seeing here is enzyme open, closed, open, closed, open, closed, open, closed, open, closed, open, closed, open, closed. This is it, on a third of a second. But we get to watch the same enzyme cranking over for a long period of time. And again, what we find is that the enzyme is highly variable. It accelerates, it slows down, it speeds up, it slows down. It accesses different conformations. In fact, it accesses at least 2 dramatically different speeds. And you can actually see that in the 40 seconds of data over here. Do you see how there's this dense region, and then a less dense region, and a dense region? The dense region corresponds to rapid switching. The enzyme has an overdrive gear that it goes into. So it flips gears to second gear, and it just starts cranking along at a much faster speed. And you can see that over here, where the enzyme is going open, close, 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 like 300 times per second. Whereas over here, it's doing open, closed, like 50 times per second. So this is dramatic. That's 6 times faster. And what's crazy is that the enzyme does this all day long. it switches between first gear and second gear, back and forth. And a big mystery in the field is what's up with second gear, okay? So to address that question, I'm going to skip some stuff. To address that question, together with collaborators, we chemically synthesized a version of the polysaccharide that didn't have crosslinks. This is like one strand of the net that I showed you earlier. Same polysaccharide, now we're using chemical synthesis to access a new substrate. And what we find is that the enzyme has a different type of activity. I do have to show you some more controls. Okay, I can't get away from this, they're crucial. These are mutant enzyme active sites. Do you remember earlier I showed you the carboxylates that are required for the enzyme to operate? So we mutated those carboxylate residues, and the enzyme no longer works, and therefore it never closes. The other one of these enzyme mutations traps the covalently bound form of the substrate that I showed earlier in the ping pong mechanism. And the enzyme never can hydrolyze back off the substrate. And again, it never gets back to closed. Okay, so I get to finally tell you what the difference is between first gear and second gear. This is a day in the life of an enzyme. Here's how it spends its time. Okay, so this is an enzyme cranking along happily, being fed either the linear substrate or the cross-link substrate. In the case of getting the cross-link substrate, it gets to hydrolyze things about 50% of the time. But if you feed it the linear substrate, it goes wild. It gets to hydrolyze glycosidic bonds 88% of the time. And so, this over here is second gear in blue. That's non-productive rapid chatter. And over here in the cross-linked, the blue is much more apparent. So in other words, the cross-linked substrate, the substrate found on the surface of the bacteria cell, corresponds to second gear. So what we think is happening is the enzyme is mowing across the surface of the cell, chewing contentedly, bond after bond after bond after bond, happily hydrolyzing it all. And then it hits one of these peptide cross-links and gets stuck. And when it gets stuck, its response is to start chattering away. It flips gears, and it just starts going 6 times faster. And what we think is happening is that it then transits along the peptide down to the parallel polysaccharide. So in the same way that DNA has a 5' directionality and a 3' directionality, polysaccharides have a directionality as well. And it turns out the surface of the bacteria cell is a highway of parallel polysaccharides. So the enzyme comes along, hits a cross-link, goes down, zooms along, hits cross-link, goes down, zooms along, down, across, down, across. So what the enzyme is doing is zigzagging across the surface of the cell as it chews apart the surface of the bacteria. And in retrospect, this totally makes sense because, again, the enzyme evolved to poke holes in bacteria. And by doing a 2-dimensional rip in the surface of the bacteria, this makes the enzyme much more effective at killing its bacterial targets. Questions? Yeah.
So, one of the activity you see is the actual catalysis. The other one is it transporting itself.
Yeah, it's a non-productive chatter that we think is at moving along one of these peptide cross-links. So, yeah, Anthony.
So as soon as it encounters something it can cut through, it'll switch back over.
Until it finds a new glycosidic bond, and then it goes to town again. Okay, well, let's move on. I want to talk to you about other enzymes. We have lots to talk about. I want to talk to you very briefly about proteases, which cleave amide bonds. We've seen these. We've seen examples of these. In blood, there's a whole cascade of proteases that are used to respond to damaged blood vessels with a series of factor 7A, factor 10A, et cetera, proteases where one cuts one and the next one cuts another, next one cuts another, et cetera, all the way down to the point where you get production of fibrin, which then could cross-link to replace, to fix the damage up here. Okay, so this is an important mechanism for blood clotting. And naturally, if you're missing any one of these proteases, or one of these proteases happens to be mutated, you're in big trouble. Your blood will not clot. This happens in inbred families, such as the royal families of Europe in the turn of the century. This is the Tsar Nicholas II, whose wife Alexandra passed on the gene for hemophilia to their son Alexei down here. And again, this is a mutation to either factor 9 or factor 8, which are both X-linked genes. These are genes that are found on the X chromosome, so they're passed along by the mother. Okay, apoptosis is also regulated by a series of proteases. Apoptosis, the cell suicide mechanism that we talked about earlier in this quarter. And each protease activates the next one. So you have a protease up here called a caspase that cleaves the next caspase in line, which then cleaves this, et cetera. So this guy cleaves this guy, which cleaves this guy, et cetera. Let's take a closer look at an example of a protease. The protease I want to show you is one of my favorites. It's isolated from one of my favorite fruits, papaya. And it's perhaps appropriately called papain. Because it's isolated from papaya. It's an example of a cysteine-based protease. And it's furthermore another example of a nucleophile-based enzyme mechanism. And I chose this one because cysteine, of course, is the preeminent nucleophile, as illustrated earlier today when we talked about engineering in a single cysteine on the surface of the enzyme as a way of attaching it to a specific spot on the carbon nanotube. Okay, so here's the mechanism for how this enzyme works. In practice, it's actually a fairly complicated mechanism. It's a complex mechanism in the sense that it's a concerted mechanism. Specifically, here's the nucleophilic thiol functionality of the cysteine in the active site. I don't think I pointed it out here, but here's the cysteine. That's the business end of the molecule in its active site. And in a concerted mechanism, the cysteine is simultaneously deprotonated to attack nucleophilically the amide bond, and then this amide bond carbonyl gets protonated by an acid residue that hovers above the carbonyl of the amide bond. This happens in one fell swoop, from nucleophile to protonation, all at once. And that is really, that kind of concerted dance of catalytic efficiency is yet another example of what makes enzymes so special. The fact that everything is kind of held together at once lowers the transition state energy, right? So now you don't necessarily have to stabilize a protonated carbonyl in an active site. Instead, you wait until electrons appear up here on this oxygen before it gets protonated. So that lowers the transition state energy for this transition state. Okay? There's other ways of depicting this as well. I prefer this one, concerted mechanism. So all of these, okay, actually I'm going to skip the serine-based proteases. They're similar to the cysteine-based protease I showed earlier. I'm going to skip the zinc proteases as well. There's quite a few others. Like the kinases, the proteases, because they're involved in crucial processes in human physiology, such as blood clotting, which you would not want to happen, here and there, because these are involved in such crucial processes, the enzymes themselves are tightly regulated, oftentimes regulated by some loop in a pro-enzyme that has to be cleaved, where the terminology of pro means a reaction takes place that then converts it into the active functionality of the molecule. So for example, prodrugs are precursor drugs that are then converted into the active drug by some enzymatic process. And over here, we see a proenzyme that has a loop blocking access to the active site of the enzyme that gets cleaved, and then that allows the proenzyme piece to get to dissociate and turn on the enzyme. So enzymes can be very readily inhibited. You can do things like have transition state analogs. We've talked about transition state analogs before. Here is the transition state for hydrolysis of an amide bond, and here is a very effective phosphoramide transition state. Notice that this also has the tetrahedral geometry of this transition state up here. And if you do that, you can actually very effectively inhibit this enzyme. Other types of inhibitors Phosphonates down here, phosphoramides over here. These KIs are the dissociation constant for binding, where it's KD, except it's for inhibiting, binding and inhibiting the enzyme. And again, smaller numbers equals more potent enzymes. And notice what a champion phosphoramidite is with a picomolar, near picomolar inhibitor. Okay, now there's a million other things I could talk to you about. I'm going to pick them up next Thursday. When we come back, we'll be finishing off chapter 6 and going on to chapter 7. Midterm will cover through today's lecture.